If fitness is more a matter of persistence than replication, what does this say about the nature of hereditary memory? Well, it says a lot of things. Perhaps the most important thing it says is that hereditary memory operates on a much larger and more diverse scale than the typical Darwinian conception of hereditary memory residing in the gene. The gene selectionist concept of evolution points to DNA as the principal locus of hereditary memory. Richard Dawkins' book, The Extended Phenotype, says this explicitly. The subtitle of the book is The Gene as the Unit of Natural Selection. So too does the genetic theory of natural selection, which supposedly rescued Darwinism from the mutationist threat of Thomas Hunt Morgan. It did no such thing, of course. What it did do was to bolster Morgan's mutationist conception of evolution. This focus on the gene is modern Darwinism's most vulnerable weak point, and for two reasons. First, it's a very narrow conception of hereditary memory. And second, as we learn more about DNA and how it works, our concept of the gene is undergoing substantial revision. As an aside, it's also the most vulnerable weak point for the most prominent criticism of Darwinism today, namely intelligent design theory. So, let's have a look. A few lectures ago, I spoke of how the central dogma of molecular biology bolstered the idea that natural selection of genes was the only conceivable way evolution could work. This argument was based upon a deep parallelism between the genetic theory of natural selection and James Watson's and Francis Crick's revolutionary new understanding of the structure of DNA. The central dogma basically states that there is a one-way flow of information from nucleotide sequence code in DNA, which is transcribed into ribonucleotide sequence code in messenger RNA, which is then translated into amino acid sequence code in the form of protein. Because DNA's sequence code can replicate and be passed on to future generations, DNA not only specifies function, it constitutes hereditary memory that is embodied in genes. Specific nucleotide sequence codes correspond to specific proteins, and messenger RNA, for its part, is a form of scratchpad memory that is used to connect function and structure to hereditary memory. Genetic determinism is the obvious implication of this. Function and structure are specified by DNA nucleotide sequence code. The central dogma thus aligns very closely with the genetic theory of natural selection, what we might call the central dogma of modern Darwinism. In a population, a gene might be represented by several alleles, indicated here by the various colored circles. These determine particular functional states, or phenotypes, that are specified by the sequence codes in the alleles. These functional states might differ slightly, be specified slightly differently by the various alleles. Some of these functions may not be well suited to the environment where the bearer of the allele lives. That is to say, they will specify inapt function. The bearers of these inapt function alleles will not reproduce as prolifically as others whose alleles specify better suited, more apt function. Competition is thus between apt function alleles and alleles that are comparatively inapt. In this conception, evolutionary change can only come about through some mutation that may produce a phenotype better suited to the environment, better aptitude, in short. This notion of a strict genetic determinism is nothing new. We've already encountered Thomas Hunt Morgan's mutationist theory, which was quite dogmatic that the only possible form of hereditary memory resided on the chromosomes. He was equally dogmatic in his assertion that evolutionary change could only come about through mutations in those genes. Gene determinism did not start with Morgan, however. At the end of the 19th century, gene determinism was bolstered by this man, August Weismann, to whom we were introduced a few lectures back. Weismann, remember, reconciled Mendel's particulate theory of inheritance with the developing realization that the hereditary stuff, whatever that was carried on the chromosomes. But Weismann is perhaps better known today for his own argument for genetic determinism in evolution. His argument is based upon a rhetorical device that has come to be known as the Weismann barrier. Weismann, like all biologists at the time he lived, was consumed with the problem of soft inheritance versus hard inheritance. We spoke about this problem when we were outlining the crisis of Darwinism in the late 19th century. Go have a second look if you need to refresh your memories. The crisis was how, or whether, adaptation in the organism could somehow be transmitted across lineages. This was the principal point of departure that Darwin tried to draw between himself and Lamarck. Lamarck, for all his flaws of details, basically was asserting that the mechanisms that drove adaptation in individuals could also drive adaptation in lineages. This idea stuck in Darwin's craw, and he was determined to set himself apart from it. But as we saw from his theory of pangenesis, he was never quite able to do that. Enter August Weismann. Weismann was a developmental biologist. He studied how embryos develop from the single-celled zygote. In development, he saw a kind of evolution at work. In fact, the word evolution historically meant the gradual change of form and function during embryonic development. The organism consisted of cell lineages descended from the zygote. The embryo evolved in the classic usage of the term from the zygote. The different tissues that arose during development were due, Weismann thought, to a kind of soft inheritance that was passed along the zygote's cell lineages. Everything started anew with each generation. New zygote, new embryo and adult, new adaptation, and new evolution. Whatever soft inheritance did during embryonic development died with the organism, and with it the soft inheritance that drove development. By this logic, adaptation during an organism's lifetime could not be passed down to future generations. How then to explain evolution? Weismann invoked a new concept for the time, the germplasm. It was through the germplasm that traits were passed from generation to generation. Why the offspring of, say, chickens were always chickens and not, say, sparrows. Weismann's germplasm was a form of hard inheritance in his view. Soft inheritance, where it had a place in Weismann's thinking, was a form of embryonic inheritance. But Weismann's theory of the germplasm and soft inheritance turned out to be completely wrong. Weismann's theory turned on something he called segregation of the germline. Here's how germline segregation is supposed to work. The germline passes through the gonads, the testis and the ovary. These give rise to the sperm and ovum, respectively, the gametes, in short. At fertilization, these combine to produce the zygote. Following fertilization, the single-celled zygote starts the process of cleavage, dividing first into two cells, color-coded here as yellow and green. These also divide, producing 4 cells, color-coded again. The embryo will be either male or female, depending upon the genetic sex of the embryo. And germline segregation occurs when these cells' descendants develop into the testis or ovary of the new organism. I've color-coded these to reflect their descent from either male or female. The descendants of the other cells produce the soma, or body. In other words, all the cells that make up the tissues, organs, and all the rest of the organism. In this way, there's a very tight continuity between the germ cells of one generation, let's call it generation 0, and the germ cells of the next generation, let's call it generation 1. It's through modification of the germline that evolution is supposed to occur. Whatever changes that are brought about in the cells of the somatic line disappear with the death of the individual. Germline segregation means that the cells that give rise to the germline are segregated from the somatic lineages very early in embryonic development. August Weismann's concept of germline segregation profoundly shaped how we think about adaptation and, in turn, how we think about both Darwinian or Lamarckian evolution. To reiterate, gametes in the form of sperm or ova combine to form a zygote. Very early on in embryonic development, two types of cell lineages emerge: the cells that will give rise to the body, or soma, and the cells that will give rise to the germline. Even though the germline and somatic lines compose the organism, Weismann argued that there had to be a hard barrier between the two, that the germline and soma were strictly segregated from one another. What emerges from the soma is the body of the organism. Let's symbolize this with a mollusk shell. This woodcut, by the way, is from one of Cuvier's catalogs of the mollusks of the Paris Basin. Take a few moments to look at this cartoon and familiarize yourself with its features, including the serrations along the lip, the pattern of fluting, and how tight the spiral of the shell is. The adult organism, the soma, can adapt, remember? And this means that the form of the adult body can change according to new conditions. Take a moment to compare the serrations, the fluting, and the tightness of the spiral. This is organismal adaptation, and there's no question or controversy that organisms can adapt. However, the idea of germline segregation means that adaptation within the organism, the somatic line, will not be transferred to the next generation. Why? Well, the next generation arises from the germline, from the sperm and ova, which combine to establish a new organism. This germline is segregated from the somatic lines, and the result will be a next generation that looks very much like the previous generation, descended from a newly established somatic line. Same pattern of serrations along the lip, same tightness of spiral, and the same patterns of fluting. In other words, germline segregation imposes a kind of reset of form from one generation to the next. And the only way a change in a lineage can come about is through some mutation in the germline, signified here as a change of the color coding. The assumption of germline segregation imposes a particular way of thinking about evolution. If we follow an imaginary phylogenetic tree through time, evolution proceeds by continuity of the germplasm from one generation to the next. In other words, it's only through changes in the germplasm, again indicated here by variation of color, that lineages can evolve. The somatic lineages, by contrast, are ephemeral, and because any adaptation in the somatic lines cannot affect the germplasm, the story of evolution becomes solely the evolution of the germ lineages. The concept of the Weismann barrier fits in very nicely to the whole concept of the gene as the sole unit of natural selection. Whatever happens in the organism, however the organism adapts during its lifetime, is limited to the organism itself. This cannot be passed on to future generations. Changes in the germline are the only way evolution can occur. Because of this, Weismann's idea has been adopted as support of the gene selectionist model for evolution. Mostly, it's been treated as a decisive refutation of the Lamarckian idea. The correspondence is a little too convenient, though. Because it's so convenient, quite a bit of legend, historical revisionism, and uncritical thinking has come to surround Weismann's idea. For example, what about his supposed disproof of Lamarckism? Among the hoariest of the legends is that Weismann experimentally disproved Lamarckism so decisively, in fact, that no credible scientist could ever again advance the idea. Talk about convenient. It conveniently left gene selectionism and mutationism as the only viable ideas left standing. Weismann's supposedly decisive experiment involved chopping off the tails of several generations of mice. If Lamarckism was correct, then mice several generations hence should be expected to have shorter tails. Weismann's result? There was no observable change in tail length, despite the lineage having had its tails repeatedly shortened in each generation. Proof, right? This interpretation only works, however, if you think Lamarckism is the inheritance of acquired characters. As I've argued in previous lectures, that is not the essence of Lamarckism. Weismann's experiment is more a refutation of Darwin's theory of pangenesis. There are a host of other problems with the Weismann barrier. I've outlined these more fully in this video. For example, germline segregation can only apply in organisms that have germlines. This does not include plants, bacteria, protists, and a host of other non-animal creatures. These evolve too, and without segregated germlines. How do Weismann's ideas apply to them? There's also the complication that even within animals, germlines are not always so well segregated. We'll be getting into this in a subsequent lecture. The most serious problem, however, is a persistent misrepresentation of Weismann's motivations and his reasoning. Weismann's whole motivation was to build a model for the interplay of soft inheritance and hard inheritance during embryogenesis. His whole idea of segregation of the germline was a logical necessity of that model, which turns out to be false, and on many levels. If you want to learn more, you can read about it in my most recent book, where I outlined Weismann's theories in some detail. The Weismann barrier is therefore more of a barrier that wasn't. Virtually none of its assumptions, or the narratives that have been constructed from them, turn out to be correct. But the Weismann barrier is convenient to one of those narratives, the genetic theory of natural selection. Never mind whether the foundation is sound or not, it has been shaped to support the narrative. The emerging story of hereditary memory and evolution is turning out to be much richer, in fact.